Thank you, Ralph, and thank you everyone for being here this morning. I have to start sharing my heartfelt congratulations to Kathy, who I feel like I've been connected in a special way to you. I started my fellowship the year you founded the IVTA, and I still remember the first time we met at ASCO. And I will always cherish that moment because you made a big impact on me. So thank you very much and congratulations again. All right, well, welcome. I hope you are all energized. We have a very exciting program this morning. We designed the agenda with the idea that it should be interactive and informative, and we have several intimate breakout sessions addressing very important aspects. We have sessions dedicated to navigating the stages of care, including treating an active tumor, survivorship, and what to do when a tumor comes back. And we have also other breakout sessions dedicated to thriving with a brain tumor, and that will include exercise, nutrition, fertility preservation, and also a very special focus on caregivers. So I predict you're going to have a very hard time to choose what sessions to attend, but thankfully all of them are recorded, so you can watch them later, which is what I will do. So please look at your program book or find all these sessions in the Whova app for a list of all the breakout sessions and where to go. And after these breakout sessions, you will also hear from a 3-time caregiver about building resilience. And later, we will have a neuroscientist giving us an intriguing lecture about how the brain makes decisions. So all of that will be later today, but this morning we will be starting with a session focusing on clinical trials. And I think this is especially meaningful this year. So a reminder, first of all, to our virtual attendees to go back to the agenda and click on the link for this session. So, in this session that we will have now, we will talk about finding better treatments. So, in this session, you will learn how a new drug is developed and approved to treat a brain or a spine tumor. And this will include the ins and outs of clinical trials, the drug development process, as well as what happens after a drug is found to be effective. And for all of this, we are very, very lucky to have Dr. Jennifer Clarke. Dr. Clarke is a neuro-oncologist and clinical trialist at the University of California, San Francisco. She focused on gliomas and targeted treatments, as well as immunotherapy. And in her talk, again, you will hear about all this amazing information about drug development. And she's also going to use the example of borisadenib, the drug that was recently approved for treatment of low-grade glioma. She will take that as an example and walk us through that process so we understand it better. So without any further delay, I'm going to introduce Dr. Clarke. Thank you, Dr. Clarke.
Good morning. It's a real pleasure to be here today to be able to talk with you. I'm going to just take a moment and make sure I'm set for, all righty. Perfect. Okay. So I'm going to talk through the process. It's quite a long process of going from clinical trials all the way through to actual drug approval. I do need to disclose that I have both consulted for Servier and have also received research support for clinical trials from them. But I am going to be talking about vorasidenib as an example as we go through this process. So how does a drug get to market? There's a really long process here that starts well before the clinical trials. So clinical trials are where I come in for the most part, but it really starts a long way before. And so there's drug discovery, there's then drug development, moving into preclinical testing, clinical trials, FDA drug review, drug approval, and then even after that, the job's not done and there's post-market monitoring. Starting from the beginning with drug discovery. So what happened with IDH mutation? These were first discovered back around 2007, 2008. And what actually happened on the left, there was a group of people who were looking at molecular evaluations and genetic mutations in glioblastoma. And what they realized was that about 12% of glioblastoma patients had a mutation in a gene called IDH1. This mutation tended to be in people who were younger and in people who had what was at the time called a secondary glioblastoma, meaning a lower grade tumor that got more aggressive over time. And they realized that these people also did better in general. And so that was certainly an interesting point. And that led the second group on the right side of the slide to start looking in the lower grade gliomas, grade 2 and grade 3 gliomas. And they found that mutations in that IDH1 gene were present in around 70% of grade 2 and grade 3 oligos and astros, and in, again, those secondary glioblastomas, as they were called at the time. And they further realized that the people who had those tumors who didn't have an IDH1 mutation, a number of them actually had a mutation in a similar gene, IDH2. And so that became further intriguing. I don't want to scare you with this slide. This is a schematic of the metabolic cycle that goes, that most cells go through in generating energy, and you don't need to worry about the majority of this. What I want to point out is that as part of this process, isocitrate gets converted to alpha-ketoglutarate, and that's what the IDH1 and IDH2 protein enzymes are responsible for. The alpha-ketoglutarate moves on to be part of the energy generation process, and that's great. That's what you want to happen in most of your cells. If you have a mutant IDH protein, though, what happens is some of that alpha-ketoglutarate gets shunted off and is instead converted into something called 2-HG. This is a metabolite or a product of this metabolic cycle, but it's not actually a useful one. And it only shows up where these mutant proteins are in the tumor. So that's called an oncometabolite. And so the question then became, well, we're pretty sure we don't want that around. And so what happens if we're able to block those mutant proteins and prevent the development of the 2-HG? Does that screw up the ability of the tumor to live and grow? And there were two drugs that were developed. One was an IDH1 inhibitor called ivosidenib. The other one was an IDH2 inhibitor called, even we have trouble with these, I gotta say, enasidenib. And so that was a good start, but the question was, could we actually do better than that? And we weren't really sure how well these two drugs actually got into the brain. And so moving into the second step now, the drug development, there's this candidate category of drugs, and what they did was they took this initial compound that has a number, it's a fancy chemical schematic. This was a compound that actually fit very nicely into this little pocket in the mutant IDH1 protein. And so that was a good starting point. And so from there, what they did was they said, okay, so we're going to kind of systematically make changes to this structure and see if we can come up with something better. You don't need to worry about all the numbers and things here, but what they did was they took these two spots in the molecule that are labeled with an R, and they started changing what the compounds were that were attached at that point. And the way that this works, these are different models. The little numbers here, the smaller the number, the better the binding and the better the fit. And so they went through one by one and looked at what the binding was and made further changes. And what they ended up with was what was called AG881 and eventually became called vorasidenib. And the numbers were very low and the binding was very good. Even more importantly, though, vorasidenib bound nicely both to IDH1 and IDH2, and so that meant it could really be used for any of the IDH mutant tumors. You didn't have to worry about knowing exactly which mutation was there. So that was exciting enough to say, okay, now we're going to move into the preclinical testing in the lab where we've committed to this particular compound, and now we're going to see kind of how well does this really do. And so there were some cell culture studies, but then there were mouse studies where we really wanted to see how does this work in a living entity. And the way that this is done is using what's called an orthotopic mouse model. And what orthotopic means is that the tumor that was implanted in the mice was actually implanted in the brain. Because again, part of what we care about and part of what's so hard with brain tumors is, does a drug actually get into the brain and into the tumor? And so this is a complicated graph. What this shows is these dashed lines. The green one on the bottom is the amount of 2-HG that is in normal brain tissue. So it's very low, which is what you would expect. The gray dash up here is the level in an untreated IDH mutant tumor. So it's very high. So the question then is, what if you give vorasidenib to a mouse with this kind of tumor? And what they found was that the level of 2-HG dropped way down to this red line here. So basically indistinguishable from normal brain. And so that was pretty exciting. That's really what we want this drug to be doing in a tumor. And the mice also did okay with this compound, didn't have some unreasonable toxicity. So that led us to say, okay, we're going to now try this in real people. Clinical trials are a whole kind of lecture on its own. Normally that's the lecture I give. I don't talk about this beginning and end stuff. But now that we have a drug that's gotten all the way through, it's fun to talk about the entire process. So for clinical trials, the first part are phase 1 studies. These are what are called dose-finding studies, usually very small, less than 30 patients, trying to figure out what's the best safe dose that we can give to people. Once we have a dose that we think works, we move to phase 2, where we're really, again, looking at safety of that specific dose, but we're also starting to look at, is this potentially effective, and is it going to do what we want it to do? These are still small studies, but if something looks promising there, then that's where we move to the big, exciting phase 3 studies that are the definitive comparative trials. We're comparing our test agent against some standard treatment. They're almost always randomized, they're frequently blinded, and this is where we're in the hundreds to thousands of patients range. So phase 1, again, the focus here is safety and toxicity. I'm going to give you some vocabulary here. We are looking for major safety issues, major side effects that are called dose-limiting toxicities, or DLTs. And the goal is to find the highest safe dose that is below a certain rate of DLTs. And that safe dose is called the maximum tolerated dose, or MTD. The goal and the general kind of assumption in oncology studies is more is better. It's a very kind of classic American approach, I think. So, dose levels on the right here, probability of something happening is on the left. And for the blue, the assumption is the more drug you give, the better the effect is going to be. But we also recognize that the more drug you give, the more likely there is to be toxicity. And so what we're trying to do is we're trying to hit this sweet spot in the middle where we're having good effectiveness, but an acceptable level of toxicity or side effects. The other goals that we have with phase 1 clinical trials are to really learn more about the drug and how it works in the body. And we're looking for what are called pharmacokinetics, or PKs. That's the level of how much drug is in your bloodstream at a certain dose and how much gets into the tumor at a certain dose. Even more importantly, we're looking then at something called pharmacodynamics, PD. And that is, if the drug's in the tumor, is it actually doing what we want it to do? In this case, is it inhibiting that 2-HG production? And we look at all of those different factors, the effectiveness, the side effects, the PKs, the PDs, and kind of come up with what we think is the right dose to move forward We call that the recommended phase 2 dose. Most phase 1 clinical trials are done in a pretty simple way. It's called a 3+3 design. What we do is we enroll 3 patients at the first dose, and if nobody has any significant toxicity, that's great. We move on to the next higher dose level and enroll 3 patients there. If 2 or 3 patients of those have a DLT, then that's obviously not a great situation, and so we stop and we de-escalate down to a lower dose. If 1 out of those 3 patients has some toxicity, then we're in this gray zone. It could be that the drug's starting to cause some problems, or it could just be that we really honestly had 1 unlucky person. And so at that point, what we do is we add 3 more patients at the same dose, and we see how they do. And if none of those 3 have issues, we dose escalate. If any of those patients have issues, we stop and we de-escalate, and we go from there. So it's sort of this decision tree that we go through. For vorasidenib, we started at 25 milligrams as the first dose level, and this is a once-a-day dose. That went okay, so we went to 50, and then we went to 100, and 200, and all the way up to 300. And there were some little stars there. We didn't really have major issues with dose-limiting toxicities, meaning big side effects, but we were starting to notice that there were some bumps in the blood tests monitoring the liver. And so these liver enzymes were kind of starting to go up as we got to these bigger doses. And so it was enough at the 300 milligram mark that they went back down, and then we kind of went back down to 100. At that stage, based on what we were seeing with the liver enzymes and the information that we had about the drug pharmacokinetics at that point, the decision was actually 50 milligrams is probably enough, and we might even be able to get away with less than that. And so there was a question of whether or not we wanted to think about a dose as low as 10 milligrams. The point being that you don't need the biggest dose, you just need the dose that's big enough to do what you need it to do. And if you can get away with a lower dose with less toxicity, then that's great. So those were the doses that were selected to move forward. Now we're going to phase 2 trials. This is again where you're getting more information about safety and toxicity, but you're also then starting to look at efficacy or effectiveness. Because these are still small trials, they are only moderate power, meaning that you can't see small changes, you can only see relatively big changes. And we try not to have these studies go on for years and years and years. And so we use what are called surrogate outcomes, meaning instead of looking at somebody's actual overall survival, which can take a really long time, we look at progression-free survival, or how long do we control the tumor. In the case of vorasidenib, we also needed to know, is this really getting into the brain? And so what was done next was a surgical study, and it was decided to look both at vorasidenib and ivosidenib, which is the IDH1 inhibitor we had talked about way back. And this was a study that was designed for people who had IDH1 mutant gliomas, where the tumor was growing and we thought that surgery made sense, but it wasn't an emergency. And so what we did then was we did what's called a window of opportunity study, where we would treat people with the drug for about a month before surgery. What that lets us do is then we can take some of the tumor that's removed and we can look at, does the drug actually get in there, and is it doing what we want it to do? And so, and then after surgery, all of those patients are allowed to continue on with drug, and that gives us more of the kind of phase 2 looking at effectiveness opportunity. So this was appropriately published as a phase 1 study, because we're still doing those PK, PD things that I was talking about, but there was a phase 2 part of it as well. This is a complicated surgical schematic that may be a little hard to see. I'm going to talk you through it. What we did then was patients with the IDH1 mutant tumors, because ivosidenib was one of the drugs, who were needing surgery, enrolled. We treated them with, we randomized them, and in the first stage of the study, people were randomized between ivosidenib at 500 milligrams daily, vorasidenib at 50 milligrams daily, Or we had a small number, 5 patients, who didn't get any drug before treatment. Those were our control from the standpoint of making sure that we could see that there was 2-HG production in the tumor if we weren't giving the drug. We also had what's called an external control, where there were a bunch of people whose tumors were already removed and stored in a research tissue bank, and we went and looked at the level of 2-HG in those tumors as extra information for untreated folks. These people then, once they had been treated for a month, they went to surgery. And the plan was, if we saw that we were actually blocking the 2-HG successfully here, then we wanted to look at some other doses. For the vorasidenib, we wanted to go down to 10 milligrams and see if that was enough. For the ivosidenib, they wanted to look at a different schedule. So instead of 500 milligrams once a day, we looked at 250 milligrams twice a day to see if doing that more kind of continuous dosing was actually better. So everybody in these pre-surgery groups, once they got to surgery, after they recovered, they were able to continue the dose that they had been taking of the drug, or those 5 lucky people got assigned to one of the 2 drugs afterward. And then everybody was treated until the tumor grew. This is some of the numbers for the PK, the drug levels in both the tumor and the plasma. And again, you don't need to worry about these detailed numbers, but the important take home here is we got tissue successfully from 47 out of 49 of the patients. That's pretty good. And 42 of those samples were evaluable, meaning that we got enough tumor to be able to look at, and that they had been taking the drug the way they were supposed to be taking it before the surgery, so we knew what we were looking at. And so all 5 untreated patients plus 37 treated patients had samples that we were able to look at. And what we saw was that in general, on this left side, the levels in the tumor were very good compared to the levels in the blood or the plasma. Here is the pharmacodynamics for the study. And again, I'm going to talk you through this. The gray is the untreated tumors, either in the study or in our external control. And what you can see is that the level of the 2-HG is quite high in those untreated patients, which is what we expect. The patients who were treated with vorasidenib, especially that bigger 50 milligram dose, we saw a very nice suppression of the 2-HG, so it went away. And we also saw that, although it's not quite as tidy, in the ivosidenib in both groups. So it looks like both of these drugs do get into the brain and do what we want it to do by suppressing the 2-HG. So then what happened with the people in the phase 2 part after the surgery? This is called a swimmer's plot. Each lane or row is an individual patient who is treated after surgery. The dark blue is the 50 milligram Vora, the light blue is the 10 milligram Vora, and then this is the 500 and 250 twice a day of the IVO. This is time, and so we're out all the way to 2 years here. What you can see is that many patients actually were treated for quite a long time. And all the people with these little triangles at the end were actually still on treatment at the time that we collected the data. Not everybody did well on the drug, but an awful lot of people. It looked like we controlled things for quite a while. And I think one of these folks in one of these swimmers' lanes is actually sitting in the audience right now. So that led us to these conclusions. So both drugs had good brain penetrance, and they did suppress the 2-HG, which is what we wanted. The ivosidenib 500 milligram daily looked pretty good. The vorasidenib, especially the 50 milligrams daily, looked very good. And both of the drugs had pretty reasonable safety profiles still, and looked like they were really controlling things, potentially for a meaningful amount of time. So that was enough of a phase 2 result that we said, okay, we're going to bite the bullet and we're going to phase 3. And so this is the INDIGO study that I think many of you have heard quite a bit about. What we were targeting for the INDIGO study, because now you have to really think about what's the patient population that we think this drug really makes the most sense for. There had been all kinds of different pre-treatments in the people who were in the surgical study, so it's a little hard to know. And what we thought was that it made the most sense to treat people who had grade 2 tumors, obviously with the IDH mutation, who had had only surgeries, and where there was some residual tumor left on the MRI that we could see and we could watch. We were prioritizing people where we were concerned. Is it growing? Is it not? We're not totally sure. But we wanted a group of people where we didn't feel like we needed to commit to a more aggressive treatment like radiation and chemotherapy. So we were comfortable potentially kind of sitting on things. And so those patients were enrolled. They were randomized, and this was a blinded study, meaning half of people got the vorasidenib at that 50 milligram dose, half of people got placebo. And then we watched with MRIs every 3 months. If we got to a point where we thought there was tumor growth, we were allowed to unblind and find out what the person had been receiving. And if somebody had actually been getting the vorasidenib, then we obviously needed to move to a different treatment. But if somebody had been in that placebo group, the protocol, the study, did allow for us to then treat those patients with the vorasidenib in an open label, meaning we knew that that's actually what we were giving them. The primary outcome for this study was called PFS, or progression-free survival, but we also were looking at overall survival and looking at quality of life and cognitive outcomes and seizure control as well. So this is what's called the Kaplan-Meier curve for the results, and I have to tell you, this was pretty exciting. So what we see here is time going across, and this is the number of people who have not progressed. And so over time, the red line, which was the vorasidenib group, clearly did better than the placebo group. And the median progression-free survival was about 27.5 months versus 11 months. That's a big difference. And so that was pretty exciting. We looked then at a bunch of different subgroups. I know you can't read anything that's along the side, but this is looking at different age groups, different gender, different geographic locations where people lived in the world, because this was a worldwide study. We looked at where the tumor was in the brain. We looked at if it was an oligo or an astro. And basically, got too excited. Any of those subgroups, it looked like the vorasidenib did better than the placebo. This is another way of looking at outcomes. This is called time to next intervention, meaning how long before we felt like we had to actually do something different, like radiation and chemo. And again, you can see that the vorasidenib group was much longer than the placebo group. So that then led to what's called a new drug application, where the drug developer has to put together this giant package to send to the FDA. The point of this is to show that the drug is both safe and effective for the intended use in the population that you've studied it in. And you really have to include everything in this packet. So not only from the preclinical data in the lab to the clinical trial results, but you also have to include if there were any clinical trials done outside the US, even though this is for US approval, they want to know. You have to include all the safety updates. You have to include who you think this should be given to, and information about which patients you think make the most sense. Once that's submitted to the FDA, they look at it and decide if they feel like it's a complete enough package for it to be worth their time. Assuming they do, then there's a review team that goes over it, and there's multiple different members of the review team. Each member reviews the pertinent parts. So there's scientists, there's pharmacists, there's clinicians, there's radiologists, and so on. They each review their part. The FDA also sends what are called inspectors out to some of the sites that participated in the clinical trials, usually some of the ones from the phase 3 clinical trial. And they look at all the data, like the source data, from where everything comes from. And what they're doing is they're making sure that it wasn't sloppy, that there weren't errors, that people didn't deviate too much from what they were supposed to be doing. Generally just that there were no red flags, that there was something fishy going on that makes them question the data. There's a project manager who then pulls all that together into an action package, and the whole review team comes up with a recommendation for what they think should happen. That's given to a senior FDA official who then actually makes the decision. And the decision can be, yes, it's approved. It could be, no, it's disapproved. Or they could say, you know what, we actually need more information. We want you to tell us this, or we actually want you to go do another clinical study and find out about this. So you could imagine if this hadn't already been done, the FDA might have said, you know what, you got to show me that this actually gets into the brain. In the case of vorasidenib, it was complete enough that, as you know, it did get approved a few weeks ago, and so that was pretty exciting. But the job's not done there. So once the drug's approved, you have to figure out what is the labeling or prescribing information, and there's a whole bunch of specific stuff that has to go into that. You've seen those package inserts that you get with every drug you buy, either from the pharmacist or over the counter, right? And so you have to specify what is the indication, meaning what's the clinical situation that you think the drug's most appropriate for. You need the dosing, obviously. You need to know about the potential side effects and warn people about that. And then you need to know about the necessary safety and monitoring precautions. And so these are pieces that I pulled from the actual package insert from vorasidenib. So it was a bit of a last minute talk, you can imagine. So again, I know it's pretty hard to read this, so I will read to you. The indication for vorasidenib is for the treatment of adult and pediatric patients 12 years of age and older with a grade 2 astro or oligo with a susceptible IDH1 or IDH2 mutation after surgery, whether it's been biopsy, subtotal resection, or complete resection. The dosage is the 50 milligrams daily that we talked about from all the studies. They shifted the formulation along the way so that it was a little bit more manageable to produce. And so the equivalent in the new formulation is 40 milligrams, but that is the 50 milligram dose that we looked at in all those studies. The most common adverse reactions that they saw were fatigue, headache, COVID-19. These weren't all necessarily related. Muscle pain, diarrhea, nausea, and seizures. They also saw elevations in those liver blood tests, and in a few patients they saw a drop in what are called the neutrophils, or a particular kind of white blood cells. So based on all the information they had, as far as the safety monitoring and precautions, definitely don't recommend this in someone who is pregnant because of the concern for fetal toxicity. For most people who are taking the drug, the primary thing that we need to watch is the liver blood tests. And we check those every 2 weeks for the first 2 months, and then monthly for at least the first 2 years, depending on how somebody is doing. We're still not done because there is this post-market monitoring that I mentioned at the beginning. So the FDA appropriately has realized that, you know, by the time you get to this approval point, you've given the drug to a fair number of people, but a fair number of people in this case is on the order of like 500 to maybe 1,000 people. That's not that many in the kind of scheme of the overall world, right? And so once a drug gets approved and it starts getting given to tens or hundreds of thousands of people, there may be safety issues or logistics issues that come up that you didn't run into in that very select group that you were watching super carefully. And so there are ways to report issues that come up. There's something called MedWatch, which is a web page where clinicians or patients can report that they've had a side effect or a toxicity from a medication, and it's a place where the FDA can post new safety-related information for existing drugs. There's also something a little more proactive than that was implemented a number of years ago called the Sentinel Initiative. So you know that there's giant amounts of data these days that we collect and that we're able to better analyze than we used to. And so this Sentinel Initiative is looking at huge electronic databases, things like electronic health record systems, insurance claims, and so on, and basically looking for people where this drug was prescribed, is there any kind of trend or concern that's popping up more commonly in those patients than in other people? And so these are ways that we can keep an eye on patients as drugs get out into the real world and make sure that we're not inadvertently causing toxicities that we didn't know about. So that's the end of the journey for now. Thank you very much for your attention, and I am happy, to take questions, even though I don't have a slide that says questions. There you go.
Thank you for that presentation. Obviously, a lot of excitement and hope with vorasidenib, but unfortunately, it's not a fit for everyone with ogliodendrogliomas and astrocytomas. So I'm wondering if you can speak to perhaps the hope that those of us that can't use the drug should have. Is this maybe a leading indicator of research in the field of other gliomas that perhaps will be more similar treatments down the line for a broader spectrum of use cases?
Yeah, so that's a great question, and there's kind of several important pieces that pop into my mind. So the first thing is that IDH mutant gliomas are a somewhat unusual subset where we think this is really one of the very first mutations that happens that's very critical for the development of these tumors. And that means that these tumors are in general fairly sensitive to messing with the IDH mutation. It really hits the tumor hard. And so other tumors where there are more complicated combinations of mutations, we haven't found one single bullet that kind of hits as effectively. We have to be more creative, and that's why we are, for many other clinical trials and lots of other research, we're looking at a combination of different targets, trying to hit them all at the same time to see if we can actually take out entire tumors. The other situation where this is not necessarily a great fit is people whose tumors that have the IDH mutation, but they have been around for a long time, they've gotten more aggressive, they're growing more quickly. This on its own is probably not going to be enough to knock that down. I didn't really focus on it as I was talking, but that surgical study, the patients who were eligible for that, the tumors had to be mostly non-enhancing, meaning looking less aggressive, because the drug hasn't looked as promising in more aggressive tumors. But that doesn't mean that it doesn't necessarily, that it might not make sense to try combinations there also. And so there is an ongoing study right now for enhancing IDH mutant tumors, looking at the combination of vorasidenib and an immune drug called pembrolizumab. And the hope is that doing some kind of combination like that might hit the tumor harder. So those are a couple of the directions that we are going to try to better address some of those concerns.
Hello. Sorry. Thank you, Dr. Clarke. Just as a reminder, you can also submit questions using the app. For our virtual attendees, please type and submit your questions in the Q&A section, and we'll try to address your questions as well. So we do have a question here on the app, Dr. Clarke, that is asking, if you are on a clinical trial, can you ask to get the data from the trial that you're on, or do these patients have to wait until that information is published?
That's a great question. The answer is yes, you can certainly ask. There is some information that is available kind of as we're going along, where we're getting a sense of how much toxicity there is and those sorts of things that certainly can be shared in real time. And in fact, we're obligated, if there are significant new risks, to let people who are on these trials know, because they need to know whether or not, they need to decide whether or not they're comfortable continuing. The final results of does it really look effective generally aren't available until the whole trial is done and has been analyzed, and that's kind of after the fact. And similarly, some of the analyses like those PK and PD tumor tissue level kinds of things, we don't do those in real time for each individual patient. We collect all the tumor and they kind of do it all at once so that we're hopefully doing it consistently. And so that sort of information is not necessarily available in real time either.
Just a question from this side of the room. Hi.
Hi.
Thank
Sorry,
you
it's a little hard to see.
Sorry. Thank you for the presentation. Really excellent information, and I appreciate the way you broke it down for a non-clinical audience. two kind of questions. One is, can you say which other mutations are now being potentially targeted by other drugs, or is there anything promising out there? I know this kind of piggybacks on the gentleman's question from up here, but what's sort of next potentially on the horizon as far as mutations and specific targets go, either in isolation or in combination? And also, I didn't quite understand why the researchers decided to go with VOR versus the IVO. Is there a reason why one was better than the other? I just didn't quite catch that. My husband received the IVO, but I think it was quite late in the process for him, and the tumor had been non-enhancing, but it was a grade 4 because it had multiple mutations, but then it turned enhancing. So I think, unfortunately, this was all coming about a few years ago as his tumor was becoming much more aggressive.
Got it. Okay. Good questions. So I don't know if I can go back for my slides or not. But the slide where I was kind of showing you how much the 2-HG got blocked in the tumors, the 50 milligram vorasidenib one, it was this skinny little box, which means that it was very consistently inhibited across the way. Let me see if I can get back to that quickly. Okay, so can I do this? So the fact that that was very skinny meant that it was just sort of all the drugs at that, or all the tumors at that dose were nicely inhibited. Here, what we're seeing is overall they were well inhibited, but there was a little more variability from one person to the next. So the two reasons I think that the vorasidenib was the one selected to go forward were, one, that this was very nice and consistent, and then two, that advantage that it could block either IDH1 or IDH2, so you just don't have to worry about which of those mutations you're dealing with were the reasons. As far as your other questions, so there are a number of other mutations that we actually have been looking at for some time. For a relatively select subset of people with gliomas, tumors have what are called BRAF mutations, and there are some excellent drugs that target that particular mutation that actually have been approved in certain particular conditions. In glioblastoma, one of the common mutations that we see is called EGFR. And so there have been a bunch of drugs that have been looking at targeting EGFR. We haven't found quite the right approach to doing that yet. And I think because that's not one of the earliest mutations, it's a little further down in the chain of events. It means that not every cell in the tumor necessarily has the same EGFR mutation. And so I think that's still a very attractive target for us to aim at, but it's not enough to do it by itself. You have to do it along with other things. And so looking at combinations in that kind of setting is one of the things we've been doing. I don't know if you all know, but glioblastoma in the past was actually called glioblastoma multiforme. That's the M in GBM. And the reason it was called that was because the cells are so variable, both how they look under the microscope, but also with the genetics and the mutations from one cell to the next. And so that's part of why glioblastoma has been such a challenge, is because there's so much variability.
Hi. I was diagnosed with glioblastoma in 2018, and I've been stable on a clinical trial since 2019 with ONC201, and I was curious your opinion on that drug.
That's
and what
wonderful.
You've seen with it.
So that is a drug that has looked potentially promising, particularly, and this is another specific mutation I should have mentioned. There's a class of tumors that are called diffuse midline gliomas that have a mutation in something called H3K27. And in some glioblastomas, but also particularly in people with diffuse midline gliomas with that mutation, the ONC201 drug has looked pretty promising. And so that is being tested in particular in people with those mutations, hoping that that's going to turn out to be an effective treatment. What I can say for you is, heck, if you've been on that since 2019 and you're good, I would stay on it.
All right, we have a question here on the app about the Indigo trial. Can you talk a little bit about how the Indigo trial measured for quality of life?
Great question. So there were a number of surveys that are pretty well standardized that were given to patients asking about symptoms and about quality of life. And they were asked before they started the treatment, and then they were asked periodically along the way. And my gestalt understanding of the results was that people's quality of life was pretty good taking the medication, but those analyses are still being finalized and should hopefully be published within the foreseeable future.
Thank you, Dr. Clarke. So as you know, I'm a 6-year patient starting my 7th year next month on this wonderful drug. And just to mention that I was able to participate in a webinar last week with my neuro-oncologist, your colleague, Dr. Jenny Clarke, Taylor, pardon me, too many Jennys.
Yes.
Even my wife's name's Jennifer. And that's exactly what we talked about, was not only the mechanism of the action of the drug and all the rest, but also I was able and privileged to be able to talk about my patient experience, quality of life. So that's going to be released sometime in the next week or two. So those of you who are interested in that, all you have to do is go to the UCSF website. Thanks.
From the horse's mouth, if you will.
All right, I have another question on the app here asking about standard of care. Does a patient need to have had completed standard of care to qualify for a clinical trial? Or if they are currently on standard of care, does that disqualify them from participating in a trial?
Excellent. Okay, so this is a very good question. In general, the approach for clinical trials is for one of two situations. One is if the tumor has grown back after standard treatment, and in that case, in order to be eligible, you do in fact have to have done whatever is considered to be the standard treatment, and then your tumor grows back and you're potentially eligible for that type of clinical trial. There are also clinical trials that are designed more for what we call the upfront setting in the initial diagnosis. Indigo was, roughly speaking, one of those trials where the people who were eligible had had only surgery and no other treatment before enrolling on the study. There are many clinical trials for other tumor types also, including glioblastoma, where there is something potentially extra added on top of the standard treatment that either starts right at the beginning of somebody's radiation therapy or sometimes starts with kind of after the initial radiation, potentially with or instead of the Temodar that people normally take during standard of care. And so if there's that type of clinical trial, then you are potentially, in fact, eligible before you actually go through the standard of care, although it's often you will be getting that standard of care as part of the treatment on the clinical trial. I think if somebody is kind of midstream where they have been on standard of care and things are okay and the tumor doesn't look like it's growing, then you're generally not eligible for a clinical trial at that point. The idea being, if what we're doing is working, then we should stick with it, and we don't think crazy and we don't move to something different and experimental unless that stops working. Did that all make sense? Okay.
All right, I think I see a question here.
As somebody who runs clinical trials for brain tumors, how do you account for maybe other things patients may be doing in the background, from exercise to nutrition to alternative therapies? Obviously, we don't want to prevent patients from trying things, but we also don't want to contaminate the trial. So how do you think through these issues?
That's a great question, and also a very complicated one. So I'm not going to say this is the right approach, but I think it's the practical approach that we mostly use these days. If there is something that is an officially recognized treatment for tumor, then we obviously don't want somebody to be doing that in addition to whatever we're doing within the trial. But if there are other things that people are doing, including more holistic interventions of exercise and diet and over-the-counter supplements, we really can't control for that, and we don't. So people can choose to do what they think makes the most sense for them. And you're right that does make the clinical trials potentially messier and harder to know exactly what the impact is of whatever we're testing is. But that's the reality of testing things in real people, right? The mice in the lab, we can control everything. Can't control the sleep, I guess, and the activity even there. But for people, there's a ton of variability, and this is what makes clinical medicine in human patients. It's what makes it messy, but also amazing.
Would you like patients to report to you if they are doing these things?
Would I like them to report if they are doing these things? Generally speaking, yes. Most of the time, the clinical trial has set up kind of, here are the things that we specifically want to ask about and document. And so we will ask about those, but it is in general always a good idea to let your doctor know whatever it is that you're doing, just so that we have the most complete picture possible.
All right, I think we have time for just one last question. I think it's a good one to end on. Can you share how patients can proactively research clinical trials?
That's a very good question. So I think the first and foremost, talk to your medical team and ask at each step along the way, is there a clinical trial that you know of that might make sense for me? Because there's a million trials out there, and it's really hard to keep track of all of them and to know which ones might or might not be appropriate for your individual situation. So that's part of our job, is to help filter some of that out for you. If you're wanting to be proactive beyond that, there's a site called clinicaltrials.gov that is a list of all the different clinical trials in the country, if not the world. And it's a daunting list, but you can search by specific things like grade 2 oligo or glioblastoma that's recurrent, or other specifics, and start to narrow down the list of potentials. I think realistically, depending on where you live in the country, it makes the most sense to look in your general region for what trials are available. As a general rule, you have to go to a site that has the study open and get the treatment from them and follow up with them and work with them. And so it's repeated visits over time. And so if you're living like me in California and there's a clinical trial in Hong Kong, that doesn't make a whole lot of sense, right? And I'm not sure that I would want to uproot myself and move to Hong Kong to live there to do a clinical trial there, because I've lost all of my social network, my support, the rest of my life, right? Which is what we're doing this for. And so kind of balancing those aspects is an important piece. And I think really, again, the most important part of this is talking with your home clinical team about what makes sense and whether there are trials that potentially are a good fit for you.
Thank you so much for that great advice, and thank you all so much for your questions. We're going to move forward with the rest of the program.
Thank you.